but some of you might be interested in going to grad school to do research and to get a PhD. So the key point, which is very different from medical school, is that if you are a PhD student in biology or biomedical engineering or biochemistry, etc., you get paid instead of paying. So you get about a stipend of about $25,000 a year, and you pay no tuition. So that's not so terrible. The focus is on research, and so what that means is that if you want to get into graduate school to do research, you have to do research now. And why is that? Because if you go and you apply, coming from UCI, where there are more than 200 labs doing research, if you go and apply to a program saying, oh yeah, I'm really excited about research, and you have no research on your application, you're going to look at that and say, I'm a frustrated med student, denied. So yeah, if you think you really are interested in research, you've got to actually do some and figure out whether it's true. The more you do, the better, because the more you do, the more the people whose labs you're in will be able to write complimentary things about you, hopefully, and give real content. So if you want to get into grad school, do research. And the way to do that, basically, is to do 199. You can contact people. Go through websites, figure out what you're interested in, and contact them. If you have trouble, come talk to me. I'll be happy to help you. But it's not very hard. The key thing is persistence. People are busy. I often get distracted and don't respond when people email me, but if they email me 2 or 3 times, I'll generally respond. So, don't worry about being too aggressive as long as you're polite. The worst that will happen is people will say no. Okay. Any questions for that? Okay, so there's a lot of material that we're going to be talking about. And so you need some foundational questions that you ask over and over again to help you make sense of all of this. And so I'm giving you here a few of them. So one set of questions has to do with the creation of organization or self-organization. So cells aren't a big bag of chemicals. They're doing things. They've got a lot of structures. They've got all these different organelles. They've got compartments where things are going into or out of these compartments. So you should be asking, how do a group of molecules assemble into the cell? How do you form a mitochondria? How do you form a lipid droplet? How do you make sure that the proteins that are on the lipid droplet are different from the proteins in the nucleus? Right? How do you control all of these things to make sure that all of these subassemblies have particular functions? So associated with that have to do with which properties of the molecules or which interactions are important for a particular function. And then how do you tune those to regulate the process and to change what's going on? Associated with creation and maintenance of organization is the cytoskeleton. The cytoskeleton can be used to move and position things. And so how does that work, and how does that interface with other aspects of the cellular program? Then there's the whole question which comes up over and over again about regulation. So it's sort of amazing that a cell can do things, but even more amazing is that the cell can change what it does in response to particular signals, either environmental or developmental signaling. And so what processes are changed and how do they change? So in particular, how does the cell know what to change? So how does the cell, at a molecular level, suppose a cell is infected by a bacteria and is going to respond to try and kill the bacteria, how did the cell know that a bacteria was there? Right? It doesn't have any eyes. What is it that it detects? Right? And then once the cell has detected that it wants to change somehow, what are the set of molecular interactions that it controls to do so? And how does it do that? And then finally, sequestration or compartmentalization are often used to control processes. So you should always be thinking about that. So as an example, cells can commit apoptosis. They can self-suicide. And that's often very useful and important to avoid a pathogen taking over or to avoid cancer spread. So how does that work? How does the cell control that it, say, undergoes suicide or not? Well, for this case, things like cytochrome C that control apoptosis are sequestered. They're kept inside of mitochondria, and then they're released response to certain things where the mitochondria has decided, all right, it's time for the cell to kill itself. So the mitochondria will release cytochrome C, starting this whole process. So that's an example of sequestration where you choose to do different pathways depending on what's allowed to be in the cytoplasm versus controlled inside a subcellular compartment. There are many such examples where controlling what goes on involves controlling what's outside. Okay, so that brings us back to microscopy. So this is a pretty complicated diagram, and I don't really want you to know too much about it, but there are a couple of key features that are important for microscopy. So for fluorescence microscopy, the idea is that a photon comes in, and that's this green line here, and gets absorbed by the fluorescent molecule, and when it does so, the fluorescent molecule jumps from its ground state to its excited state. Okay? And that photon that came in had a certain amount of energy, and then almost immediately afterward, about 10 to the 7th to 10 to the 9th seconds after that photon is absorbed, the molecule that's in the excited state jumps down to the ground state again and emits a photon, which is this guy in red. Okay? So the key thing is that the photon that's emitted has slightly less energy than the photon that was absorbed. Okay? And for light, the amount of energy that's in the photon is inversely correlated with, or inversely proportional to the wavelength. So shorter wavelength is higher energy. So if you, for green fluorescence protein, for instance, as a fluorophore, you shine in 490 and it absorbs 490 nanometer wavelength photons, and then when it emits, the photons are 509 nanometer wavelength. So they're slightly longer, which means that they have slightly less energy. Okay? And so the whole trick for fluorescence microscopy is how do you excite with 490 and detect at 509 and not detect all of the stuff that's 490 that's very close and would also look like green to your eye. Okay, so how do you separate these two? That's the whole thing that we're going to talk about next, which is where filter cubes come in. So this is a sort of a simple diagram of the filter cube. And the idea is your light source is over here, and this is your light source from your microscope that is going to basically go into your sample. And it's going to excite fluorescence in your sample, and then your sample is going to fluoresce. So the idea is you're shining 490 nanometer light into your sample. The light is absorbed by your green fluorescence protein. It's going to emit at 509 nanometers, and so that light is ultimately going to be detected by your detector. But what you want to do, what this filter cube is going to do, is it's going to make sure that your 490 gets to your sample, but that none of the 509 that was present initially in your light source is going to get to your sample, because that would confuse you. And that all of the 509 that comes from your sample gets to your eye, but that all of the 490 that hit your sample and scattered off doesn't. Okay, so the idea is the filter cube is what's going to filter between the light that you're exciting with and the light that is being generated at the sample. So we're going to talk about how that happens. So there are three elements, optical elements, that comprise this filter cube. The first is called the excitation filter, the second is called the dichroic mirror, and the third is called the emission filter. And so the light goes through them sequentially. So your light source is here, and your light source will usually be either a laser or a mercury arc lamp, which is very broadband. But in either case, often the light source that you're exciting with is going to have both the wavelength of light that you need to excite your fluorophore, but there's also going to be some contamination from a little bit of the wavelength that your fluorophore is going to emit. Okay? And so the first thing this filter cube has to do is it has to prevent the additional light from that's the wrong wavelength from getting to your sample. And why? Because if this light that is 509 gets to your sample, it could just scatter back off of the sample and come to your eye, and it's going to look like it's being generated, but it's going to be generated randomly on your sample just from the reflection, so it's going to give you a lot of background. So you're going to get a lousy image, but you're going to have a lot of background. Okay? And so you could sort of think about this as if you're in a parking lot and there's a light on inside the car and you're trying to see that, but there's a lot of light hitting it and shining off of it from, say, the sun. You're never going to see the light that's coming from inside the car because there's all this background. Okay. So that's what you're trying to eliminate here, all the background. So how does it work? So this first filter here is what we call a short pass or a filter. And so this filter is going to transmit anything that has high energy that is below 500 nanometers in wavelength. So anything above 500 nanometers in wavelength is going to reflect. So any light that is 509, which is the light that's going to be generated by your sample, doesn't get through because it's reflected by the excitation filter. Okay. So the light that comes in that makes it past the excitation filter is 500 nanometers or below. So that's what you need to excite your fluorescence. Okay, and then that light basically hits this dichroic mirror. So di is 2 and chromic is color. So there's a 2-color mirror. And what that means is that it responds differently as a mirror to different colors of light. So in this case, this is a long pass. So anything that's long goes right through it. So anything above 500 nanometers goes right through it and isn't reflected down. So anything that's 509 that happened to somehow magically make it through here doesn't go through, because it goes straight through rather than reflecting down. But anything that's below 500, since it's not passed, since it's shorter, it's reflected. So this is a mirror for 500 or below, so the 490 sees the mirror and gets shunted down into the sample. Okay? So now you're exciting your sample with a lot of 490, but no 509, hopefully. Okay? Now, the light that's in your sample excites your fluorophore and then gets emitted at 509. Now, some of the light that was 490 bounces off of your sample, and you don't want to see that because that can confuse you. So what happens is, remember, this is a long pass mirror, so anything that's less than 500 it acts as a mirror for. So anything that bounced off the sample that was below 500, like the 490, hits this mirror and is reflected back. Okay, so this is a mirror for all of your excitation light. It's a mirror. Okay, but the emission light, the light that's 509 that was generated by your sample, that basically, this mirror is opaque, is transparent to that, so that light goes right through. So that light basically goes up and up and up into your detector. Okay. So then the question is, well, what does this emission filter do? And this emission filter is basically almost the same thing as your dichroic. It has the same properties where it's long pass and it lets the 509 through and it reflects the 490. And so it's basically just an additional layer of selection to even larger reduce any 490 that came from the cell. So combined, these three allow you to see a lot of the majority of the 509 that was generated and not see any of the 490 that was used to generate. Okay? And so this is the filter cube that would work for GFP, because we talked about these and their specific wavelength cutoffs. You can understand that for different fluorophores, like red fluorescent protein that has different excitation and emission, you need a different filter cube. Okay. So in practice, when you're using fluorescence on a microscope, you will have a slider with filter cubes, and you will slide the right one into position into the light source, and you use that one. And depending on what fluorophore you use, you're going to need a different filter cube. Yes? That's right. This is to see the fluorescence coming from the protein that's marked. That's exactly right. Okay, so that was sort of standard fluorescence microscopy, and that just shows you the distribution of the protein that's marked inside of the cell. Now, we can also start to ask more sophisticated questions about whether the protein that you're interested in is interacting with another protein that you're interested in. So are the two proteins close together or not? So we have protein A and protein B, some generic proteins. So we have A here, and then we have B. And the question is, how far apart are these proteins? And so what you can do is you can attach GFP to A and RFP, which is red fluorescent protein, to B. Now, if you were then to use two colors of light simultaneously, you could see some green dots and some red dots from where the proteins were in the cell. Okay, and that wouldn't tell you anything about their interaction. That would just say that both proteins are there. But it turns out that there's this process called fluorescence resonance energy remember, there's a different wavelength that's required to excite the green fluorescent protein from the wavelength that's required to excite the red fluorescent protein. So the green, say, is excited at 490 and emits at 509. But it turns out that you can find some proteins, for instance, maybe the red, which is excited at 509 and then maybe emits at 535. Okay? So what you can do is, it turns out there's this process where if the guy that's emitting is close to the other guy that can accept it, that there's a transfer of the photon from this guy to this guy, as long as they're close. So instead of emitting the green, what happens is you're exciting at 490, but the only thing you see coming out is at 535, because the energy has been transferred over. Okay? But that only happens if these two fluorophores are very close together physically. And by very close, I mean within about 20 nanometers of each other. Okay. Yes? Is that what we call bleeding? No. Well, no, bleeding in general is where you've got the spectrum of fluorescence is a little bit broader. So when I, for instance, say that this emits at 509, it doesn't only emit at 509. There's actually a width of the distribution. So it's from between about 500 to about 530 or something, right? So and the peak emission is at 509. That's where it emits most, but there's sort of additional wavelengths that it emits at. And if you have two fluorophores which have different peak emissions, some of their spectrum of emission overlaps. And that's called, you can then get bleed through where you detect one as the other. Okay, that's different from this. This is where you designed it so that when they get close, the energy from one gets transferred to the other guy fluoresces. And so what you can do is, by measuring the amount of emission you get at 535 when you excite at 490, you can measure how close these guys are together. So if they're far apart, if you excite it at 490, you're only going to see 509, you're going to see nothing else. But as they get close together, the closer they are, the more what you observe is when you excite at 490, you emit at 535. Okay? And so you're seeing this transfer of energy, and it allows you to measure the actual physical distance between these guys. So the way you would use that is you would have proteins A and B, and maybe you think that under some conditions these proteins bind to each other and interact. And so if you had that model and you wanted to test it in the cell, suppose that when the cell cycle starts, you think that one of the first things that happens is that these guys form a complex. So what you would do is you would put into the cell DNA coding for protein A, GFP, and DNA coding for protein B, RFP, and then you would look for fret signals where you're exciting at 490 and detecting at 535, and when in the cell cycle they came together, if they did, you would get signal. And so then you could prove that they're coming together only at specific times during the cell cycle. So it enables you, inside of the cell, to measure the association of proteins with nanometer resolution. So it's kind of amazing. So this takes, a pretty specific model for these two proteins interacting, and you're really going to the work to label them and get them into the cell. It's a lot of work, but it enables you to ask and answer very specific mechanistic questions about what's going on. Okay, so what I want you to get from microscopy, what I've talked about, is I want you to know how a filter tube works, and I want you to know about labeling proteins via antibodies and immunofluorescence, and by fusion proteins with GFP. Okay, and then I want you to understand the basic idea of fret and kind of when you would use it. My goals for that. Questions on that? Okay, so what you get from microscopy is sort of where something localizes, and you can see if you're using GFP, which allows the cell to be alive, you can see how its localization changes over time. And you can get something about the number of, say, mitochondria or the number of things. You can see individual blobs. But you often can't get the absolute concentration of a protein, and you can't often get the function of a protein. you can sort of see where it localizes, and that can give you some hints, but it doesn't tell you exactly what its function is. And certainly, sort of mechanistic questions often are not addressed. So to address those things, you need to do biochemistry and other approaches. So that actually brings me to the first of a set of approaches that have been used to figure out what proteins do. So if you take a step back and you put yourself in the 1950s, where we knew kind of that there were proteins, but we didn't know what any of them did. Okay, and you ask, how do you figure out what it is that a protein does? Okay, I mean, turns out that they didn't know then, but there are thousands of them, right? And so it's sort of a daunting task, since mostly you can't see any of them, and it takes a lot of work to study one of them. It's mostly a pretty daunting task to figure out what they'll do, which is why, for instance, we don't understand so much yet about cancer and controlling it, right? Because there are lots of proteins involved, and they often have multiple roles. So when I say, how do you figure out what each one does, often proteins don't have a single role. Often they do 2 or 3 things. Right? But sometimes what we mean by what does a protein do, or what is it important for, has to do with what does it do for a particular disease. What's its role in development of Alzheimer's disease? So there we can sort of flip the question and say, suppose you're interested in Alzheimer's, what are all the proteins that contribute to development of Alzheimer's? And then how do you figure those guys out? So one approach that was incredibly powerful and continues to be powerful is a mutational screen. And so the idea is, suppose that you can design a particular experiment where you can recognize a particular phenotype, which is some physical effect that is related to what you're interested in. As an example, suppose you're interested in sight. You don't know any genes or proteins that are involved particularly in sight, but you'd like to understand the set of genes that are controlling sight. So if you want to understand sight, one thing you can do is you can use a model organism like flies. So it turns out that many of the same genes and proteins that work in flies work in people, and you can do all sorts of things in flies that you couldn't do to people. And so one thing you can do is you happen to know that if you take your hand and you threaten to squash a fly, it will fly. Right? You know, they have a flight response. And so in order to do that, obviously they have to see your hand coming. So then you can design a screen based on that, where what you do is you feed the flies some chemical mutagen, and that will cause mutations in the DNA, which will cause alterations in some of the proteins. And some of those alterations in some of the proteins are going to impair their ability to see. Now, some alterations in some of the proteins are going to do other things, but some of them are going to impair their ability to see. And so then, if you do the same experiment on some of these flies, what you're going to discover is that some of the flies, when you threaten to squash them, they're not going to do anything. They're just going to stay there because they don't actually detect your hand there. And so those guys are interesting. And so if you can then figure out which proteins were mutated, you may have figured out proteins that are involved in sight. Okay? And so in each model organism, there are different ways of figuring out which proteins are involved. Once you've figured out a particular fly that has a phenotype, you have to map which protein it is to mess with. And that's not so easy. It's easier now than it used to be, because now sequencing is so good that you can sequence the whole organism. But it didn't used to be the case. 10 years ago, you couldn't sequence the whole organism. So there are various mapping strategies. But anyway, you can, in fact, figure out what proteins you modified. So that helps you get clues as to proteins that are important for sight. Okay, another example has to do with cell cycle, for instance. So you would have to do, for sight, you would have to do the screen in something like zebrafish or fruit flies that have eyes. But yeast undergo the cell cycle. They divide and replicate. And so if you're interested in proteins that control the cell cycle, you can do a mutagenesis in yeast and then look for yeast that can't make it through the cell cycle. Now the trick is that some things are critical, right? If you can't go through the cell cycle, you can't divide and you die. Right? You can't make progeny. So if you hit an essential gene in one of these screens, you'll never pick it up. Because if you really mess with something that is required for cell division, you can't divide it and you can't have progeny, so you can't study it. So in fact, what people did is something really clever. And they, in yeast, they did temperature-sensitive mutations. So proteins basically are somewhat stable, but as you heat them up, they go unstable. And so you happen to know, for instance, that when you fry an egg, it changes the way it looks and the way it tastes, right? Because you're denaturing the protein. So all proteins have this ability to be unfolded by heat, but some are more sensitive than others, and some are particularly sensitive. So what you can do is it turns out that some mutations are temperature sensitive, where what happens is the mutation makes the protein marginally stable. So it works okay at room temperature, but when you shift the temperature to 37 degrees, for instance, the protein unfolds. So these temperature-sensitive mutations were critical in being able to figure out proteins that are involved in control of essential things for cell cycle. The idea was you do the mutagenesis, and then you have your cells, and you first grow them at room temperature, and then you split them, and then you have one set of cells growing at 37 and one set at room temperature. And you look for the guys that basically get stuck and can't go through cell cycle at 37, and then those are the ones that have some temperature-sensitive mutation. Okay, and then you can figure out what it is. But if you just had a mutation that totally knocked out the protein at all temperatures, you could never study it. Right? Temperature-sensitive. So I think a Nobel Prize. That was a very clever thing to do. Okay, so that's sort of genetic screens. You can do something that's similar with RNAi, RNA interference. It's also called siRNA, and we'll be talking about that. And so there, basically what you do is you introduce a small piece of RNA that double strands with a messenger RNA and causes that mRNA to be degraded. And so then you don't make protein from that mRNA. And so depending on what your RNA sequence is, you can target any mRNA for elimination. And so therefore you can knock out any protein in the cell. And so you can now robotically basically have a library of small RNA constructs which you put into cells, and you can basically knock out any protein you want in the cell. And so what people have done is, rather than saying I'm interested in this protein, you just use a robot and you go through and you knock out every protein. And then you look for specific phenotypes associated that you're interested in. So for instance, if you want, there was a very nice paper that was interested in what controls the size and formation of lipid droplets, which is something I study. So if you want to know about lipid droplets, you develop an automated microscopy tool to be able to recognize lipid droplets in a cell because you stain them with Nile red and the microscope can recognize them. And now you basically do RNAi against all these different proteins, and you look for cells where either they have too few lipid droplets or too many lipid droplets, or lipid droplets are the wrong size. And so all of the RNAis that cause one of those phenotypes have something to do with control of lipid droplets. Now you don't know what. I mean, you don't know mechanistically exactly what, but at least you now know 50 proteins that somehow contribute to the overall size and shape and number of lipid droplets. And you can do that in an automated way. It was a bunch of work, but within 3 years they knew 50 proteins, right, that had something to do with that. So that's kind of amazing. So you can do screens with RNAi, and so there are advantages and disadvantages. The nice thing about a screen with RNAi is that you know what you're knocking out, and so you don't have to try and map what you've knocked out. Okay. So associated with those kinds of screens is a sort of bioinformatic approach. So doctors see patients all the time, and a lot of those patients have disease. And so we have an ongoing experiment on the human race, right, where basically you can look for people who happen to have a particular disease and those that don't, and if you sequence parts of their genome, you can look for correlations between, genes that tend to be mutated or tend to be certain variants in people that have the disease versus don't. And so you can look for correlations between particular mutations and being prone to having the disease. And with that information, now you have risk factors, and now you can ask, so what is it that these proteins that, say, you have identified as a risk factor do? Right? And so that really is where cell biology comes into its own, because you can do all sorts of high-level correlational studies from all this information, but ultimately you need to know what it is that the protein does. And the only way you can do that is to really study it at the cellular level. And that's what cell biology does, is to study proteins and see exactly how they contribute to cellular function. So when we think about how we gain clues as to what a protein does, once we're sort of interested in it for some reason, there are a number of approaches. So on the one hand, suppose we've gotten interested in a protein and we know its sequence, but we don't know what it does. Or we know that it's important for some process, but we don't know physically what it does. So often we can gain a clue as to what it does by comparing it with other proteins of known function from other organisms. So, as an example, molecular motors like kinesins are things that walk along and drag cargoes from one place to another. And so we know what all the molecular motors are, we think, in mice, but we haven't necessarily studied them in some obscure lizard somewhere. And suppose you happen to be interested in that. Well, if you have a sequence of a particular protein in this lizard, you can do a bioinformatic comparison of that sequence with the molecular motors in mice, and if it happens to have a lot of homology, which means that most of the amino acids or many of the amino acids are the same, then you can make a pretty good guess as to the function of that lizard protein by knowing what the proteins like it do in mice. Okay? So bioinformatic approaches with homology information is very powerful. And that also works for things like you do a screen in yeast, where you find proteins that control cell cycle. And now you want to know about how the process works in people. So what you can do is, often, when you found proteins in yeast, if you do homology studies, you can associate, or you can figure out what the human homologs are. And so then you can make a guess as to the roles of these proteins in humans, even though they've never been studied, just by sequence identity. And then you can actually test that. So often, surprisingly, between, say, flies and people, there's enough conservation that if you eliminate the protein in a human cell and you replace it with a fly variant of the protein, it will still function okay. Okay, so they're often remarkably conserved, and so you can test them. Okay, so that's bioinformatic approaches and cross-species comparison. You can, of course, look at where a protein is located in the cell. So if you think that a protein might be involved in controlling transcription, right? It might be a transcription factor. Well, if it always stays out of the nucleus, it's not a transcription factor, right? Because to be a transcription factor, it has to go into the nucleus and control copying of DNA. So it's always in the cytoplasm. You know, it might control release of a transcription factor, but it itself is not a transcription. So similarly, just other things, where it's located can give you a lot of clues as to what it does, or how its location changes during certain processes may tell you something. So if it usually is focused in the cytoplasm, or is located in the cytoplasm, and then suddenly it goes into the nucleus in response to certain things, then you may have a suspicion that it helps control the response to those things. So pull downs. So the idea is you take a protein of interest, your protein A, and you want to know who it interacts with. You may not know what protein A does, but if you know that it interacts with all these proteins that control cell cycle, then it probably contributes to cell cycle. So what you do is you take an antibody against protein A, and you attach it to beads, and you make a lysate, which basically means you grind up cells. Now you put your beads with your anti-protein A antibody into the lysate, and then the antibody attaches to protein A. And now you use a centrifuge, and you spin out the beads, and along with them come protein A. Now, if protein A was bound to any other proteins, they get pulled out too. So that's called a pull down. So now you have this set of stuff that got pulled out of solution by protein A. Those are binding partners. And now you can use mass spectrometry to identify all the proteins that came out of solution with protein protein A, and those are guys that interact. Okay. And so if many of the proteins that came out of solution with protein A are related to neuronal development, then protein A probably has some role in the neuronal development or whatever it is. So guilt by association and interaction can tell you both what the protein does or what it might do, but also how it does it, right? If it interacts with proteins that control localization of a particular thing, maybe it helps control localization or regulate those proteins or something. So you get a lot of information from direct interaction studies. Okay, then you can also look at which cells or tissues the protein is expressed in at high or low doses. So if you think that the protein works to control neuronal function at synapses, it had better be present in very high concentrations in brain, because there's a lot of neuronal stuff in brain, right? And it shouldn't be present very highly in skin, because there aren't so many synapses. So you can look and see, and if it's present in a lot in skin and not so much in brain, it's probably not involved in synapse formation, right? So you can sort of figure out, based on where it's localized, kinds of roles that it might play. Similarly, if you change the environment of the cell and you change the dosage of the protein, you can get clues. So, for instance, that would sort of be environmental conditions. So, if cells respond to changing temperature by changing the proteins that are involved in unfolded protein response. So, if you heat up cells, proteins tend to unfold, and cells will express other proteins to refold them. That's called the heat shock response. Okay. So that you basically don't turn into a fried egg. It's hot. So, so if you take a cell and you stress it, and you see that the protein goes up, maybe it's involved in some sort of stress response. Things like that. Similarly, if someone shut the door back there. Oh, okay, that's cool. Okay, so you can look for correlations between groups of proteins. So we can now look at messenger RNA across the entire organism. So you can basically look at which mRNAs come up at particular times in response to, say, a disease or at certain times in development. And so if you see that when you infect the organism with bacteria, 50 different proteins go up, then those guys are probably related somehow to defense against bacteria, right? Or if you happen to know that under some circumstances, 10 proteins that are related to bacterial response go up, and then you discover that there's this other protein you didn't know anything about it, and it also goes up whenever these guys go up, maybe it plays a similar role, or it helps us. So you can look for correlations in expression levels. And that you can do at the RNA level, and there's very good tools to look globally at how RNA levels fluctuate in different responses. So, and also you can look at it during development, say in stem cells, right? You'll have groups of proteins that come up and go down together, and so you can look for correlations in those to tell you that maybe all of these guys function in a similar process. So of course, ultimately, if you alter the protein's level of function, and then you can look at the phenotypes. We sort of started with that, but you can do this in a very targeted way using RNAi. That, we talked about microscopy. So that brings us, so we've talked about microscopy, we've talked about genetics, so that brings us to biochemistry. And so what I want to end with is sort of protein gels and how they work and why they're useful. So if you take this solution of proteins, say have some cells, you grind them up, and now you've got the solution of proteins. You can't see anything. Now you can stain proteins with either plumosic blue or silver stain, and not being able to see them. But if you did that in solution, you just see that there was some color blue or some color black with a silver stain down, and you wouldn't learn anything. But what you can do is you can separate proteins by size. So you have some proteins that are small and some proteins that are big. And if you take a gel, big proteins have trouble going through the gel, and small proteins go through much easier. So if you take the proteins and you boil them in SDS, sodium dodecasulfate, which is a detergent to unfold them, and it also coats them with a unit Now if you put an electric field across the gel, the proteins are going to be pulled through the gel. They're going to migrate through, but the rate of migration depends on the size. The small guys migrate rapidly, the big guys migrate slowly. Okay? So if you put gels, if you put proteins in little wells up here at the top of your gel, so here's one well, here's another one, another one, another one, and then you let them go through, due to being pulled through by electric current, they're going to migrate, and the big guys migrate slowly, and so that's this band here, this band here, and then the small guys migrate next. Okay, so you can separate proteins by size, and that gives you a way of sort of looking at what was in your solution. Because then when you stain this this set of proteins with the Kamasi blue or the silver stain, you can see a pattern of bands, and these are individual proteins. Now, the key thing is the gel runs a little bit differently each time, depending on exactly how long you run it, so you need a size comparison to figure out exactly the size of the proteins of interest. And so what you do is you use the first lane here as a standard. It's called the ladder. And so these are a set of proteins, you know exactly what's in that ladder. And they're the proteins of known size. So you know this is 110 kilodaltons, and this is 90 kilodaltons, and this is 60, and this is 40, et cetera, et cetera. And so then when you happen to see that this guy you're interested in, you don't know what it is, but you can see it's between, say, this band and this guy, and this one you happen to know is 50 kilodaltons, and this one is 30, so this one is maybe 40 kilodaltons. So you know that there's a protein that is relatively abundant in the sample that's that size. And that enables you to eliminate a lot of proteins because you know the sizes of different proteins. You don't know exactly what it is just from seeing it. And similarly here, you know there's a whopping amount of protein that's at about the same size. So by simply separating them and then staining with a silver stain or Kamasi, you can see the individual. So that brings me to Western blots, which is where I'm going to end. So the idea is the Western blot is a two-step. It's also called a immunoblot, and it's a two-step process. First, you separate the proteins by size using the SDS beige gel. And then you basically make a sandwich, and you put the gel and a piece of filter paper below it. And you pass current through it, and that drags the proteins out of the gel and onto the filter paper. So now you have proteins on the filter paper, and they're stuck to it. And then you soak this piece of filter paper in an antibody solution. And the antibody is against only a single protein of interest. And so what's going to happen is if the protein is there, the antibody is going to bind to the protein of interest. And so what you're going to see when you look at this filter paper that's been stained for where the antibody is, you're going to see a single band that tells you sort of the amount of the protein there. So that enables you, based on the specificity of your antibody, to detect whether protein A is there or not. Okay, because it might be, and it also gives you an idea about whether protein A was high in one sample and low in another and absent in another. So that's using antibodies and Western blots. Okay, with that I will stop and we'll